It's been said that we know more about the surface of Mars than the bottom of the ocean. As interest in seafloor exploration grows, it's critical to understand life in the deep sea. NOAA Ocean Exploration is using environmental DNA as a new tool to survey the deep sea. 2 liters of seawater are collected by Niskin bottles mounted to an ROV deployed from NOAA ship Okeanos Explorer. The samples are handed off to the Smithsonian National Museum of Natural History for DNA extraction and metabarcoding using multiple assays. However, operationalizing the remaining steps in the process, the bioinformatics, data management, and data publishing, requires tools that aren't yet fully mature. Our group was tasked by NOAA Ocean Exploration to develop these tools. Precious deep-sea eDNA would be useless if we mislabeled a filter or couldn't read our own handwriting. That's why we created printable QR-coded labels that can be affixed to samples and then scanned later during processing. It's a simple solution that saves time and avoids costly mistakes. Data derived from eDNA cannot be reliably interpreted or reused if the metadata about how it was generated, key information about the methods at every step, is not reported accurately and in a machine-readable format. We teamed up with the Ocean Biomolecular Observing Network, OBON, to develop Better Biomolecular Ocean Practices, or BBOP. Each BBOP protocol has a beginning section, YAML front matter, that includes important metadata fields describing the protocol, MyOp for Omics protocols generally, and FAIRY for eDNA-related protocols specifically. Each section of the protocol is described in detail in Markdown. Documents are hosted on GitHub with version control and DOI creation through Zenodo. The YAML terms and definitions can then be recorded in a FAIRY spreadsheet, which I'll talk about next. To document all of the protocol and other metadata for an eDNA study, we teamed up with Australia's CSIRO, GBIF, OBIS, and EBI to develop the FAIR eDNA checklist, or FAIRY, built on the existing MixS and Darwin Core standards, but with additional fields required by eDNA researchers. Our group has built a FAIRY metadata template generator called FAIRY Sheets. Like the Excel template provided on the FAIRY website, the Google Sheet has spreadsheets for project, sample, experiment run, and analysis metadata. But because it's built in Google Sheets, it allows collaboration by teams. If raw DNA sequences are the code, bioinformatics is the decoder, resulting in error-corrected sequences, counts of those sequences, and species names for those sequences. Tourmaline 2 is the latest version of our amplicon sequence processing workflow. 3 steps, sequence QA/QC, denoising and representative sequence generation, and taxonomic assignment, are controlled by 3 simple config files that contain all the information needed to produce an analysis run. This example shows a real DNA sequence from an Okeanos Explorer expedition. The sequence was assigned to Macropinna microstoma, the Pacific barrel-eye fish, which I'll say more about in a minute. What is the point of discovery if you can't share it with anyone? To enable exploration, visualization, and sharing of eDNA data from NOAA expeditions and eventually any users from around the world, we created the Ocean DNA Explorer. This eDNA data portal is still in beta, but you can access it today on any computer or mobile device. It enables you to access all information about an eDNA study across multiple database tables. One of the most powerful features of the Ocean DNA Explorer is its search capabilities. You can construct complex searches of single tables and also relations between tables to find the information you want. Here we are searching for any taxonomies that contain the genus Micropinna in the NOAA OER Okeanos project. The results are cut off here, but we can see them another way. Each search result provides a human-readable sentence describing the search and a pre-constructed API query. You can copy and paste that API query as a URL into a web browser to see the results in JSON format, or paste that API query URL into your Python or R code to import the database results right into your data analysis. Using the search and navigation tools of Ocean DNA Explorer, we can see a map of DNA-based observations and other information about a taxon, here, Macropinna microstoma. Every species has an automatically retrieved silhouette image from Vilopic. It's a rapid and lightweight solution, but it doesn't always do justice to nature's amazing diversity. Check out this photograph of a real barrel-eye fish. This is not AI generated. We hope that this resource can help both scientists and the general public access and understand eDNA data better. We've created a Learn section that walks through each step of the eDNA metabarcoding process with visualizations and descriptions of the corresponding table in the Ocean DNA Explorer. We hope that these cartoons will demystify some of the jargon used in eDNA research. Additional pages within the Learn section are planned, such as search examples that demonstrate the power of the data portal. Data portals are a great resource, but they are not a substitute for a permanent biodiversity data archive. The Ocean Biodiversity Information System, OBIS, and its more terrestrial cousin, the Global Biodiversity System, GBIF, are the internationally accepted repositories for biodiversity observation data. They accept both traditional and eDNA-based data, but the resources available to prepare eDNA-based data for OBIS and GBIF are still lacking. We developed eDNA to OBIS as a tool to convert FAIRY-compliant eDNA data and metadata to the Darwin Core format used by these repositories. Taxonomic assignments to common reference databases can be mapped onto the Willms or GBIF taxonomies preferred by these repositories. eDNA to OBIS supports data from multiple assays in the same submission. All output files can be combined into a Darwin Core archive to publish on OBIS or GBIF. Finally, with all of the eDNA data from the Okeanos Explorer now processed, we can begin to analyze these results further to infer biological patterns. We are still in the early stages of this phase of the project, but we can already see distinct differences in the fish families found in the mesopelagic and bathypelagic zones. Interestingly, the mesopelagic communities are more similar to each other in an MDS space, whereas the bathypelagic communities are more distinct from each other. The next steps from this work include publishing the biodiversity observations from the first 6 Okeanos Explorer expeditions on OBIS, continuing to add features and visualizations to the Ocean DNA Explorer, and continuing to submit more datasets to the Ocean DNA Explorer. If you missed any of the QR codes in the earlier slides, you can find all of our GitHub repositories within the QR code shown here. I would like to sincerely thank all of my co-authors. The majority of work shown here was done by them, and it's truly been a team effort. Finally, we are grateful for the support from NOAA Ocean Exploration and the Northern Gulf Institute.